Today we are going to be talking on oxidase test. So before we go to the procedure, let us talk about the oxidase background. So this oxidase test is a test for the presence of cytochrome C oxidase test, and it is an important enzyme in aerobic respiration, as it is because it accepts electron as a transport chain. Now let us see the principle of this cytochrome C oxidase test. So this the ability of an organism to produce the cytochrome C oxidase test, it can be determined by using the reagent tetramethyl p phenyldiamine dihydrochloride. In short, we call it as a TMPD. So this serves as an artificial substrate donating electron. So basically that can donate some electron to cytochrome C oxidase, so the TMPD can change color. So what it does is that it has electron that can donate to cytochrome C oxidase, and when it donates the electron, it is oxidized as TMPD, that is tetramethyl p phenyl diamine dihydrochloride. And this TMPD goes colorless when it has all the electron to turning into a deep purple color. This happens when it gets oxidized, when oxidase is present to take the electron, which means oxidase is positive, or the bacteria produce cytochrome C oxidase. For example, we have Neisseria gonorrhea species, we have Pseudomonas species, we have Vibrio species. So this is the principle of the oxidase test, and let us see the reagents and the material required to perform this test. Here I have given an oxidase, the picture of an oxidase which is commercially available. So not only the oxidase test, you can also use a filter paper, dip in a freshly prepared TMPD reagent, and then here we are going to need a glass slide. So the first thing you are going to do is that you have to take a commercially available oxidase test, and then you have to pick, with the help of an inoculating loop or a choke pit, you have to pick up the colonies, the isolated colonies to be tested, and then you have to rub it gently on the disc which is commercially available, and after that you have to observe for the color change immediately within 10 seconds. You should not read your result after more than 10 seconds, because it might give you some false positive results. So here I have given you an picture of how an oxidase test looks like, but we will discuss this later on in the interpretation. So regarding on the filtered paper procedure, firstly you have to do is you have to take a filter paper that is soaked with the substrate TMPD that is freshly prepared. Then you have to moisten the paper with a sterile distilled water, then pick the colonies to be tested with a wooden or a inoculating loop or a choke pit, depends on you, and then you make a smear it in the filter paper. Now what you have to do is you have to observe the inoculated area of the paper for a color change to deep blue or purple within 10 to 30 seconds. Now let us see the interpretation. Here in the interpretation, I have given a picture of both the commercially available oxidase test and the filter paper dipped in freshly prepared TMPD. So the interpretation of the result will be same, although the procedure is different. So in the first one, here in the positive test, in the interpretation, it is written that development of deep purple color within 10 seconds. So if there is a development of deep purple color within 10 seconds, it is an indicative of an oxidase test positive. So As test sample turns the disc into a deep purple color, the test this can be reported as oxidase positive test, since the test organism has the enzyme oxidase present in them. Whereas in the absence of color, if in the site of the inoculation or rubbing within 10 seconds, if there is no development of a deep purple color, it means that the test organism does not produce any enzyme called oxidase. Therefore, the test organism can be reported as the oxidase negative. Now these methods are like very simple, and it's also very convenient to carry out. It is just a method, it just take like 5 minutes to do this procedure. To quality control, quality control here, the bacterial species showing positive and negative oxidase should be regularly tested to check the potency of the disk or the reagent. Some suggestive controls are: for the positive is Pseudomonas aeruginosa ATCC 27853, and for the negative we have Escherichia coli ATCC 25922. I hope you still remember what is ATCC, as even in my previous class I have discussed about the ATCC, that is American type culture collection. It is a type of strand to check the potency and the test organisms. So this is all about oxidase tests. Whereas there are few limitations of this oxidase test. So those are like the reagent used in the oxidase, they sometimes have been shown to auto oxidize, so hence positive results may also obtain. Whereas sometimes bacteria grown on a media containing high concentration of glucose show inhibited oxidase activity, so it is recommended to test colonies grown on media without excess sugar such as neutron agar. So this is all about the oxidase test.